everybody for joining this province-wide rounds and while enjoying the sunshine outside. Today we are privileged to have Dr. Gerry Krystal to give his talk on the role of diet in cancer prevention. And Dr. Krystal obtained his PhD degree from McGill University in biochemistry and is currently a full professor of UBC in the Department of Pathology and Laboratory Medicine and a distinguished research scientist at the BC Cancer Research Center in Vancouver. His long-standing interest is the effect of diet and exercise in both prevention and progression of cancer. He has an active wet lab at BCCRC and has published over 200 peer-reviewed papers. I'll give the podium to Dr. Krystal now for his talk. Please go ahead.
Thank you, Dr. Gao, and Sneha for saving me. Okay, so I can't seem to move the slides.
If you can let Sneha know when you'd like her to advance the slide, she'll be able to do that for you.
All right, click one more time. So you've all heard this old adage and song, what doesn't kill you makes you stronger. I would just like to add a little caveat, and that is unless it leads to chronic inflammation. Next little click. So acute inflammation is your best friend because it destroys invading microorganisms like bacteria, yeast, and viruses. And it does so in part by generating reactive oxygen species. However, next click, if it's not resolved, you start damaging yourself, and that leads to chronic inflammation, which is your worst enemy. Just about all the infirmities that plague us as we get older can be associated with chronic inflammation, from dementia to cardiovascular diseases, autoimmune diseases, and of course, what I'll be talking about mainly today is cancer. So the question is, of course, what determines your level of chronic inflammation? Next slide, please. Okay, wish I could change the slides. Okay, so of course, the next one. Next click. So your genetics, of course. So all of us are slightly different in our single nucleotide polymorphisms, just specific bases that are different. Please stop. And the ones that affect the immune system, of course, will have a big effect on your level of chronic inflammation. So for example, 2/3 of the world's population is infected in our stomachs with Helicobacter pylori. And for those who have a SNP in the promoter region of the interleukin-1 beta gene, which is a pro-inflammatory gene, are far more susceptible to developing gastritis, stomach inflammation, and then progressing to gastric cancer. So, of course, SNPs are very important. Genetics is critical. However, it tells you nothing about your lifestyle. And so the next items I'm going to mention are related to lifestyle. And of course, one of them that we're all under to a very large degree at the moment is chronic stress. So cortisol secreted from the adrenal glands, it gets your fight or flight reaction going. It increases immune cells in the blood and of course increases blood glucose to go into muscles for your fight or flight. It increases blood pressure and eventually can lead to obesity. Next, please. So, of course, history plays a big role. Unresolved wounds, sports injuries, accidents, asbestos exposure, silica, cigarette smoke, all of these will promote chronic inflammation. Next, please. Exercise. So exercise is typically associated with a reduction in chronic inflammation. Of course, as long as you don't permanently hurt yourself. During exercise, you release endorphins. You also release from your from your muscles, adenosine, which not only helps in sleep, but it also shifts to an anti-inflammatory healing phenotype, which will reduce ROS. Adiponectin, secreted from fat tissue, helps to move visceral fat, which is very bad for you. Visceral fat is the fat in your abdominal cavity. It shifts it to subcutaneous fat at the surface, which is not nearly as dangerous. You also secrete irisin, which is very anti-inflammatory. Next, please. Age and gender, of course, chronic inflammation typically increases with age, although there's tremendous variation. We completed a study looking at different ages a few years ago. Some young people have signs of chronic inflammation. Some old people do not. Interestingly, women are 4 times more prevalent to have autoimmune diseases. It's thought perhaps because the autoreactive T cells, so in your thymus, the epithelial cells produce something called AIR for an autoimmune regulatory element that helps to reduce, to kill off autoreactive T cells. It's thought to be lower in women, so they have more autoreactive T cells. Another popular theory to explain why women have more autoimmune disease is perhaps incomplete X chromosome inactivation. So the bar body that every woman, so they only have one expressing X chromosome of the 2. And because of incomplete X chromosome inactivation, they can have a heightened production of toll-like receptor 7, which promotes inflammation. Next, please. I'm happy to answer any questions at the end regarding any of these things. So the microbiome plays a huge role in chronic inflammation. It refers mostly to the bacteria in our large intestine, our colon. Very simplistically, there are 2 main phyla, the Bacteroidetes and the Firmicutes. Firmicutes tend to be tougher. Again, tremendous oversimplification. And the latter, the Firmicutes, are associated with obesity. Obese people tend to have more because the Firmicutes are more efficient than the Bacteroidetes at generating ATP, which they can give to you. So you basically, if you have a lot of Firmicutes, you can generate more ATP for yourself as well. So that can lead to obesity. The next, please. Weight, of course. So obesity, as you probably know, is the #2 risk for cancer mortality. And again, visceral fat, which increases with obesity, is a major source of pro-inflammatory cytokines. And lastly on this slide, diet, and as I will hope to convince you, has a profound effect on chronic inflammation and your risk of cancer. So the next slide, please. Next one. It's not what you eat, it's what eats you. You've all heard that. However, the next one. Yes, but it's also what you eat. So I'm just going to briefly touch on the 3 major food groups. The first one is carbohydrates, very important to talk about. So you can divide carbohydrates into simple sugars like sucrose, which is a dimer of glucose and fructose. Glucose there is a 6-carbon sugar at the top, very important role. So the starches are all glucose polymers. They can exist either as linear polymers, shown at the top, which is amylose. Now, one of the advantages of amylose is because it's linear, it can stack and form thick rope-like structures that are resistant to digestion by alpha amylase. The other form of starch that we eat is amylopectin. 70% of the starches we eat are amylopectin. They're branched, which means they're more water soluble, and the alpha amylase can get into it and cleave off dimers called maltose. In our liver, we have starch stores that are called glycogen. They're very similar to amylopectin, but even more so branched. Next slide, please. So in addition to simple sugars and starches, carbohydrates also come as soluble fiber. The three main soluble fibers are inulin, psyllium, and pectin. They are non-glucose-based polymers. So inulin, for example, is a fructose polymer, et cetera, et cetera. So when you eat benefiber, you're mainly eating inulin. In addition to soluble fiber, the next slide, there, of course, is the insoluble fibers, mainly cellulose, and that has a beta linkage instead of all the others that has a alpha linkage holding the sugars together. And apart from certain protozoa and bacteria in termites and cows, we can't break down cellulose, so that is insoluble fiber. It just gets pooped out. Next slide, please. Here we go. Now, this is one of the most important things I want you to take home from what I'm talking about today, is because resistant starch, the amylose, and soluble fiber, the non-glucose polymers, cannot be digested in our small intestine. They make it to the large intestine, the colon, where our microbiome, our bacteria, certain members, especially of the Firmicutes class, can break down, and those different colors there in the bottom slide indicate different types of bacteria. They actually break down the resistant starch and soluble fiber to what are called short-chain fatty acids. These are very anti-inflammatory. The three main ones are acetate, propionate, and butyrate. Butyrate especially is the most potent anti-inflammatory agent. It is the main food source for the colonocytes, those enterocytes lining the lumen of your bowel. They feed that. They help to generate ATP. They also increase the tight junctions that hold the colonocytes together. The three short-chain fatty acids also get into the blood, and they, again, can be very anti-inflammatory. Butyrate has been shown to reduce asthma symptoms, people with asthma, so it gets to the lungs, et cetera, et cetera. Next slide, please. And the proteins, of course, you all know about. It's just linear amino acids like beads on a string of the 20 different possible amino acids. I won't go into that. But the next one I'm going to touch on is fats, and the next click. So the major type of fat is called triacylglycerides, or TAG for short. And if you, I can point here, good. So TAGs are three long-chain fatty acids, typically shown here, attached to glycerol. So it's triacyl, acyl for the fatty acids, glyceride for the glycerol. So what happens is the hydroxyl groups on the glyceride interact with the carboxylic acids at the alpha end of the fatty acids to form an ester bond. This just shows it in more detail, and you can see typical fatty acids are just carbon and hydrogen, so they're very hydrophobic. They hate water, so they have to be specially carried when we eat them, and chylomicra, etc. So you can number fatty acids either from the alpha end, which has that carboxylic acid, or you can number it from the other end called the omega end. And that takes us to the omega-3 and omega-6 fatty acids. These are essential fatty acids, which means we can't generate them ourselves. We have to get them in our diet. The numbering is very simple. If the first double bond, so this is you lose hydrogen and you form a double bond, so it's unsaturated. If the first unsaturated bond is at the third carbon from the end, it's an omega-3 fatty acid. If it's from the sixth carbon from the end, it's an omega-6 fatty acid. So the omega-3s, again, this is very simplistic. The omega-3 fatty acids, especially EPA and DHA, are thought to be very anti-inflammatory. I'll get a bit more into their mechanism. The omega-6s are pro-inflammatory. Okay, just going to briefly talk in the next slide about how we digest these. Okay, so let's click the first click. That brings us to how we digest proteins. And so we start digesting proteins in our stomach, where we secrete hydrochloric acid and pepsin. It then goes into the small intestine, where we start digesting it in the duodenum, thanks to the pancreas producing trypsin and chymotrypsin. Pancreas is one of my favorite organs when it comes to digestion. It's very critical in providing the enzymes to help digest food. So typically, we want to digest our proteins all the way to individual amino acids, which are then taken up by the enterocytes, the cells lining the gut wall, and then they go into the bloodstream. However, certain peptides that are generating when you digest proteins are very hydrophobic, and they're more resistant to the trypsin and chymotrypsin and the pepsin in the stomach, and they can actually enter. So typically in an adult human, we don't allow peptides longer than 4 amino acids to come in. Those that tend to be longer can generate immune responses, and that's responsible for the food allergies we have. Now, in a baby, they can absorb whole proteins, and that is how babies get antibodies from their mother's milk into their bloodstream. It's because they don't break down the protein and it's directly absorbed. Now, if you have a very high protein diet, you do not break all your protein down here, and it makes its way to the large intestine, where the bacteria can break it down in a process called putrefaction, and it's as good as it sounds. You generate hydrogen sulfide, nitrosamines, ammonia, which can all damage the lining of the colon. Now, interestingly, those short-chain fatty acids I mentioned can help protect against that damage. So having resistant starch and soluble fiber in your diet plays a very important role in helping to alleviate damage to the colon from other foods you eat. Now let's go on to the next. And that, of course, is fat. Now, fat digestion is a bit more complicated because fat is very hydrophobic. And of course, your whole blood system or your whole body is based on being water soluble. So what happens here is that we make in our liver, we make bile acids, which are stored in the gallbladder. When we have a high fat meal, it makes its way into the small intestine. Nothing happens in the stomach. And the bile acids that are secreted into the small intestine act as soaps that break up the large fat droplets into little micelles, which allows the lipase, again secreted by our friend the pancreas, to help break down those triacylglycerides. It takes off some fatty acids and helps getting it into the enterocytes. In those lining of the gut cells, the enterocyte, it's repackaged into tags, triacylglycerides, together with glycerol, cholesterol, into lipoprotein complexes like the chylomicra, enters the lymph system, and then enters the blood system. So if we can go to the last one, which of course is our favorite. Now I should mention, we are addicted to carbohydrates. When we eat a donut or breads or all these things, we secrete large amounts of dopamine, and just like taking drugs. And that's because evolutionarily, it was a huge advantage to get a squirt of sugar into your system. It's very quickly metabolized, gets into your muscles for fight or flight, so it has a tremendous selective advantage. Today, of course, our biggest problem is we have too much food, where they don't have that evolutionary pressure, and these things are causing us a lot more trouble. So the carbohydrates we actually start digesting in our mouth through salivary gland secretion of alpha amylase. If you have a piece of white bread in your mouth all day, it'll start to taste a little sweet because you'll break it down to glucose. Glucose is not as sweet as sucrose. The sweetest sugar is fructose, but another story. Then when it makes it into the small intestine, our pancreas again secretes alpha amylase, helping us break down typical starches to the dimer maltose. And when we have sucrose, maltose, or lactose, it's broken down to the monomers by enzymes lining on the surface of the enterocyte. So the sugars just come in, and we'll talk about them a little more, but they make it right into the bloodstream. Okay, so let's go on to the next exciting slide. Here's your typical Western diet, which we all love. And you go into McDonald's, within 15 to 20 minutes, all these carbs, and of course, you have almost 30 grams of sugar in your Coke. Never drink regular pop. 15 to 20 minutes, you get a huge spike of blood glucose. That results in a huge spike of insulin to try and get the blood glucose down to the normal levels, which are between 4 and 6 millimolar in the blood. Okay, now, next slide. So what's really interesting here is that insulin is very pro-inflammatory. It activates and increases effector T cells. It reduces regulatory T cells, which diminish your immune response. Another click. Sorry. As well, and this is something I want you to take home, it's very important. Insulin tells the body that fuel is coming in, so you do not have to break down glycogen for your ATP. You don't have to break down fat. So when you eat a high carb meal that's easily digestible carbs, the resulting insulin tells you you cannot lose weight because you're not going to break down any existing fat or glycogen because you're going to use the fuel that's coming in to feed you. You don't. As well, insulin, it stimulates cell proliferation, which of course is what you want if you do want cancer. Next. And of course, the insulin goes to insulin receptors primarily on muscles and fat cells. So it's really good to go for a walk or a little exercise within 15 to 20 minutes after you eat to get the blood glucose into your muscles rather than into your fat. Okay, another click. So why does high blood glucose lead to chronic inflammation? Next click. In addition to what I've said about insulin, and I should just mention, going back to the top part, when you typically, when you have a huge carb meal, you overproduce insulin from your pancreas, and that causes the blood glucose to actually fall below normal levels, which makes you sleepy and hungry. And so the blood glucose drops and you eat more. So again, that's that. So, but the blood glucose, in addition to the insulin, is also pro-inflammatory. And that's because a long-term increase in blood glucose leads to what's called advanced glycation end products, or age. And it's it really means cross-linking your glucose to collagen and other proteins on the artery wall, which stiffens them, which in turn increases blood pressure, heart attacks, blood vessel damage, and strokes. In addition to that, on immune cells like monocytes, which circulate in the blood system looking for damage, those monocytes have receptors for age called RAGE, which is a great and appropriate name. So the immune cells, mainly the monocytes, recognize that age through their receptors, and they start chronic inflammation. All right, so the next slide. So how does insulin get blood glucose into mainly muscle and fat cells? And here we're talking about the glucose transporter 4, which is the main glucose transporter on fat cells and muscle cells. So GLUT4 is a glucose transporter that is what's called insulin sensitive. So here's insulin produced by beta cells from the pancreas in response to the spike in blood glucose. It goes to fat cells and it goes to muscle cells. It interacts with its receptor through the PI3 kinase pathway. It takes GLUT4, which is just present inside the cell in endosomes, brings it to the cell surface. A dramatic increase in GLUT4 at the surface helps to bring blood glucose into the cell. Through a series of 7 to 9 enzymatic steps, it's converted to pyruvate, a 3-carbon sugar product from the so you get 2 pyruvates from 1 6-carbon glucose. Here is a pivotal decision point. If your lactate dehydrogenase enzyme is active, it gets converted to lactate and is secreted. And in normal cells, that simply goes back to the liver, where it can be brought back to glucose through gluconeogenesis. However, if it makes the other decision to go with pyruvate dehydrogenase, that brings it into the mitochondria, and it goes into what's called the trichlorous TCA cycle. And the mitochondria require oxygen. So if there's low oxygen, you can't use the mitochondria and you have to go this way and secrete lactate. Now, if you have operating mitochondria through oxidative phosphorylation, you generate a hell of a lot more ATP than if you just go through this, what's called glycolysis pathway. So glucose is converted to pyruvate during glycolysis, generating 2 ATPs from a single glucose. It's either excreted or enters a mitochondria to make an additional 34 ATPs. So there's a huge benefit, especially when you're hungry and starving, to using your mitochondria to get energy. So what the last little point, one more click, is that inflammatory cytokines actually block this insulin receptor internalization. And so the increased blood glucose not going into the cells, and the increased insulin resistance. So a lot of these things have feedback, positive pathways that make things worse and worse. Okay, on to the next slide. So a critical thing happened, one more click. This was back in the 1920s. Otto Warburg, very strange man that I can talk about later, in the 1920s found that cancer cells, unlike normal cells, prefer glycolysis instead of using the mitochondria, even if there's oxygen present. Now, he thought it was because of mutations to mitochondria, so they couldn't use it, but that is a rare occurrence in most cancer cells. And most cancer cells can use both, but they prefer glycolysis. And the next slide, yeah, there we go. So the reason is, as long as you can get enough glucose, cancer cells prefer glycolysis because they can use the intermediates in glycolysis to make the fats, nucleic acids, and proteins required for rapid proliferation, and through the pentose phosphate pathway, the generation of glutathione. Glutathione is the major molecule. It's just a 3 amino acid peptide that neutralizes reactive oxygen species, which will damage DNA. So cancer cells, like the glycolysis pathway, it helps to neutralize the ROS that they generate. Okay, so interestingly, in addition to this, in the very latest issue of Cell, this is a very interesting paper came out showing that the increased production of lactate, which occurs in over 80% of cancer cells, because they prefer glycolysis, so they have to upregulate that pathway. The production of lactate leads to the lactylation, the putting on of lactate groups onto the amino acids lysines in P53. P53 is the guardian of the genome. If you have DNA damage, it tells the cell to stop proliferating. So P53 is a critical tumor suppressor that is the most often protein that is mutated and inactivated in cancer cells. So when you lactylate, put lactates on p53, it prevents it from acting as a transcription factor to turn on proteins like P21 and PUMA to stop cell proliferation. So that's a very exciting new paper. Next slide, please. So just getting back to this phenomena where cancer cells prefer to use glycolysis to keep them going and to grow. And I should mention, even normal cells that are rapidly dividing, like expansion of T cell or B cell clones, shift to glycolysis. So it's not unique to cancer cells. Any rapidly growing cell will shift to this so it can use those intermediates to generate what's needed, plasma membranes, yada, 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 for cell proliferation. So this is anaerobic glycolysis if there's no oxygen, but cancer cells use it even with oxygen present. So it's called aerobic glycolysis, or the Warburg effect. And the next little click. So in addition to everything I've said about lactate secretion in a cancer cell, so tumors typically have very poor blood vessel circulation. So when the lactate is secreted in the environment of a cancer cell, it builds up in the environment, and that lactic acid, of course, is acidic. It lowers the pH, which helps to kill the normal cells in its environment. It causes the production of proteases like matrix metalloprotease and cathepsin being released from the connective tissue, collagen, etc., fibrinogen in the environment. And that helps to increase spreading metastasis because it's breaking down, killing the normal cells, breaking down the collagen matrix. It also, the lactate production increases the production of new blood vessels, angiogenesis, to help it grow. And it, most importantly for this talk, it also depresses the immune response. It actually inhibits T cells from coming into tumors and killing off the cancer cells. Okay, so the next slide. The vast majority of cancer cells, therefore, take up and need more glucose than normal cells because they're selectively using this less efficient way of making ATP. And so you can actually detect this using positron emission tomography with the radioactive glucose analog, 18-fluorodeoxyglucose, 2-FDG. The next click just shows a person with a lymphoma, and they light up because they're taking up this glucose analog much more than normal cells. It, of course, is all piles up in the normal bladder to be secreted. Okay, so now, finally, we're getting to my work. So the next slide. So I simply asked this simple question about 15 years ago. Since cancer cells take up and need more glucose than normal cells, what if we just lower blood glucose levels using diet changes? Would that slow tumor growth? So it was a very naive question. The typical clinician would say, no way, you have to be starving before you can lower blood glucose. But we, surprisingly, in our naivety, found a totally different response. So all we did for our early studies is we lowered the carbohydrate, the next click. So we just had a Western diet, which is about 60% carb, and most of that is easily digestible amylopectin. And we made a diet where we basically kept fat the same and just switched the protein and the fat content. So we went to very high protein. And all we did in these early studies, next click, is we took mice and we gave them either a Western diet, low carb diet. We subcutaneously injected various tumor cells, and we simply monitored tumor growth rates. Very, very simple studies. Next click. Oh, and this first study is just published there, if you want to look at it. So the first thing we noticed is that we did indeed lower blood glucose levels at different parts of the day. Compared, each dot here is a mouse on a Western diet versus our low carb diet, and you can see blood glucose levels were lowered. What's interesting, they did not fall below around 4 millimolar. When you could lose consciousness, the mice were perfectly happy. Next slide, please. And next click. So we found indeed in these very rapidly going tumors that typically kill the mice within 3 to 4 weeks. So this is a squamous cell carcinoma. This is a mouse cell line. We also did it in immunocompromised mice with human colorectal cancer cell lines. And we saw a significant lowering of the growth rate of the tumor. So there's about a 3-day delay here, which is quite remarkable given these cancer cell lines are dividing so quickly. Next click. And importantly, there was no difference in the weight of the mice. Because of course, if you're losing weight through cachexia, your tumors will slow, but the weight was not changing. Next click, please. So we were mainly interested, that's treatment, but we were mainly interested in prevention. So we collaborated with Brad Nelson at the Dealey Research Center. He had these what are called NOP mice, which express an extra copy of the HER2, also called the new gene. So it's like an EGF receptor that's always on in breast cancer cells. And they lack one copy of the 2 copies we all have of that tumor suppressor P53. And these mice spontaneously generate breast cancer with about a 70 to 80% chance. So we thought, what would happen if we put mice just on these very simple diets we first came up with, a Western versus our high protein, low carb diet? And again, this is published here. The next click. We saw something very remarkable. So these mice, about 12 weeks of age, were simply put on the 2 different diets, the Western and the low carb. And at 1 year of age, so this is middle age, no mice, this of course, these are all female mice, breast cancer, none of them had any tumors, whereas almost half the mice on the Western diet did. And these mice typically live 2 to 3 years. And okay, go on to the next one, please. So then we thought, let's come up with a more quantitative model. And we came across this wonderful model called the nicotine-derived nitrosamine ketone model, or NNK. So nicotine is converted into NNK during curing of tobacco, which is just high heat and humidity. And it's activated in the liver by cytochrome P450 enzymes to something called NMAL. And if you click onto the next one, all that does is it puts a methyl group on guanosines. So with 2 rounds of replication, you've converted a GC to an AT. So this is generating mutations. So NNK is one of the 2 major types of carcinogen in tobacco. Again, for nicotine into NNK, you don't have to combust, don't have to light it up. The other major one, they're called PAHs, polycyclic aromatic hydrocarbons. Benzopyrene's a good example. You need to light it up to generate. So those are the 2 major guys. The model's very simple. The specific kind of mouse, we use females for these because they're more susceptible to lung cancer than males. They're given 2 shots of NNK after their diet change, and then they're just put in large rat cages with exercise wheels, 2-level cages, lots of exercise, because we found exercise played a big role in restraining the generation of tumors. And after 5 months, we just count the lung nodules. And I'm saying, again, the advantage of this is it's more quantitative than just a yes or no cancer. And if you go to the next slide, so here we've done a whole slew of different studies. So here we did not inject the mice with NNK, and we just asked, well, how many lung tumors do you get in normal mice where you haven't given them the carcinogen? This is our best diet, and this is the Western diet. And you can see you get 0 to 1 tumors, very, very little. When you inject NNK, the next one, you get roughly 20. Each symbol here represents the number of lung nodules. And if you start lowering the level of carbohydrate, this is simply lowering to amylopectin, easily digestible carb, from 50 to 30. And if you lower it further to 30%, now resistant, and then you go to resistant starch. When you go down to 50% resistant starch, that's what we used in our earlier studies, you get now reach a significant drop compared to on the Western diet. And we tested a whole slew of different things. Now here we're seeing a huge drop because we've introduced fish oil. So basically 30% of the fat in the 50% calories is fish oil. And you see a nice drop. If you go even further along here, more clicks. Okay, now we've replaced, oops, sorry, go back. We've replaced the milk protein casein, which is all you use here, with the plant protein soy. And that brings it down further. And we've done other studies since where we've characterized the ingredients in soy responsible for this further drop. And one more click. Okay, so if you go back just for a second, you can see when you've have a diet either with resistant starch as the 15% carb or a soluble fiber, inulin in this case, you approach the levels that you see without any NNK. So with even in the presence of this very potent carcinogen, you get a dramatic reduction in lung tumor nodules. Next slide. And this simply shows that all the mice, because they were exercising, I should add, all have similar weights. So you can't attribute the difference in lung nodule numbers to differences in weights of the mice. Okay. So now this is simply showing, and I'll explain this in a moment. When you go either, when you fast for a few days, or you go to what's called a ketogenic diet, which is very low carb, typically lower than 5%, you start switching to generating fatty acids secreted from the liver, and they in turn can make ketone bodies as well in the liver, which circulate, and you go into ketosis. And this simply shows that when you're on one of our best diets, very low carb, and the carb we have is a resistant starch, with fish oil, you go into ketosis, but you don't on the other diets. As well, and I'll touch on why this is important in a second, you also lower, with mainly fish oil containing diets, you lower a prostanoid called prostaglandin E2. So I'm just going to explain that now. Next slide, please. So here, again, once you exhaust your glycogen stores, so say you're not eating anything, you've decided to fast for a few days. The first storehouse, remember I mentioned the starch in your liver is called glycogen. It's branched, easily broken down. So you store that, and after, depending on what you read, it's a complicated literature. After about 10 hours, or it might take 24 to 48 hours, you deplete all your glycogen stores, and then you say, well, I need energy. So you start breaking down fat stored in the liver, and you release the fatty acids. You get free fatty acids, and of course they hate water, so they're transported in the bloodstream with albumin, and the fatty acids go into muscle and elsewhere for energy. They also can be taken up in the liver and converted, broken down further to these 3 short ketone bodies. And the main reason you're breaking them down into ketone bodies is because free fatty acids cannot get into the brain. They can't pass the brain-blood barrier, but ketone bodies can. So normally glucose is our main energy source for the brain. And if you go on a ketogenic diet for a few days, you have what is called brain fog because they're not used to using ketone bodies yet. And they have to upregulate the enzymes that can break down these ketone bodies back to acetyl-CoA to go into the mitochondria to generate ATP. Wonderful. The next slide. So this is just showing what I'm talking about when I talk about prostaglandin E2. So our plasma membranes of all our cells are a bilipid layer. So the little white circles here are the hydrophilic head groups that interact with the watery medium, both outside the cell and inside the cell. And then you have these 2 fatty acid chains, which as I indicated are mostly carbon and hydrogen, so they hate water. So they're on the inside of this bilipid membrane. Now, typically, one of these fatty acids can be EPA or DHA. Again, this is omega-3 fatty acids, mainly derived from fish oil, or they can be arachidonic acid. Now, this is an omega-6 fatty acid. And I should just mention that since the Industrial Revolution and the introduction of fast foods and a Western diet, we have reduced the ratio of omega-6 fatty acids to omega-3. I'm sorry, we've increased the ratio of 6 to 3. It used to be about 3 to 1, and now it's about 30 to 1. So we typically, from our diet, have more omega-6 fatty acids in the plasma membranes of all our cells than we used to have. When we get an inflammatory challenge in, say, our immune cells, the arachidonic acid is broken down, it's released, and then there are enzymes called cyclooxygenases, or COX, or lipoxygenases, LOX for short, and the arachidonic acid is converted to prostaglandins, like prostaglandin E2 and leukotrienes. These are high pro-inflammatory potential mediators in the cell, and they lead to the production of inflammatory cytokines. If instead you have high levels of these guys in your plasma membrane, EPA actually competes with arachidonic acid for COX enzymes, blocks there, and they are converted into 3 series prostaglandins, or 5, which have a much lower pro-inflammatory potential. And they can also be converted into what are called resolvins, which help healing. They're very anti-inflammatory. So you can see from this kind of slide that having high levels of EPA and DHA can reverse the inflammatory production here. Now, I have to state a lot of what I'm saying is ridiculous oversimplification. A lot of molecules, especially pro-inflammatory and anti-inflammatory cytokines we talk about, we only have a limited understanding of what they do because we could only say what they do because of the assays we've developed. Once we develop new assays that we haven't had before, we might find a lot of these signaling intermediates or inflammatory or anti-inflammatory cytokines have other jobs that we don't know about. Now, the main reason we have increased arachidonic acid and other omega-6 fatty acids, like linoleic acid in our diet, is because we've been using soybean oil, corn oil, and sunflower oil in all our oils, and they're very high in omega-6 to omega-3 fatty acids. Next slide, please. So I'm just going to touch on the microbiome very briefly. It's a collective term for the bacteria, viruses, protozoa, and fungi. And we have a huge number in our colon. It's been downgraded a bit to about 30 to 100 trillion commensal bacteria, and they're very beneficial. They weigh between 3 and 5 pounds in our colon. So if anyone says you're overweight, you can just say 3 to 5 pounds is just my bacteria. It's not me. The two main phyla, as I mentioned, the Bacteroidetes, which tend to be gram-negative, and the Firmicutes, which are more efficient at breaking down the food they get into ATP, and they give some of that to us to make us more obese. We are born sterile. We pick up bacteria going through the birth canal. The diet at an infant to dramatically influence the microbial makeup. Now, kids normally get about 10 courses of antibiotics by the age of 10. That disrupts the microbiome. It helps the Firmicutes take over because they're hardier to antibiotics than the Bacteroidetes. But again, dramatic oversimplification. There's many different classes, orders, and genus and species in each phyla. So if you take the feces from an obese person and put into germ-free mice, they gain weight. If you put them from a lean person, they don't. Diversity, which is very important in our microbiome, plummets within 10 days of a junk food diet, because with very little resistant starch or soluble fiber, that's the biggest thing missing from a Western diet, is resistant starch and soluble fiber, and your microbes will starve. Okay, as I mentioned, the resistant starch generates short-chain fatty acids, which are anti-inflammatory. And interestingly, the last thing there, next click, is that gut bacteria actually produce or stimulate from the enterocytes the production of neurotransmitters like serotonin, dopa, and GABA. And that likely plays a crucial role in autism. People with autism have a different microbiome. We don't know if it's an association or a cause, but the type of neurotransmitters that are secreted plays a big role in anxiety and depression. So that's very interesting. Okay, moving on. So we found the diets in our different mice. This is what's called a UMAP. And here we're looking at similarities in the microbiome based on their 16S ribosomal RNA sequences, and keep clicking through here. Each diet has a different type of microbiome. Please click. Keep going. So you just see they have different diets. And yeah, go forward. We found that different microbiomes were different genera, different species were associated with more lung nodules. This doesn't mean it's causal. It's an interesting association. So I'm just going to end up with, next slide, please. Just, I haven't had a chance, of course, to talk about all we've done, but just to synopsis, we found lowering carbohydrate not only slows tumor growth of injected tumor cells, but prevents tumor formation in breast and lung cancer models. We've optimized our diets. We found, for example, we've done a clinical trial giving fish oil supplements together with Stephen Lamb to heavy smokers. We found it reduces their C-reactive protein, a well-known marker of inflammation, prostaglandin E2, and the lymphocyte or monocyte lymphocyte ratio, another marker of chronic inflammation. Our optimal diet, which is resistant starch or soluble fiber here, soy, and 50%. We've also used to show that it reduces the incidence of other models, cancer models in mice, including leukemias and breast cancer, but not prostate cancer, which fits with early stage prostate cancer in humans being triggered by fat, which they use as a food source rather than upregulating GLUTs and bringing in sugar. But we also are just finishing up a study where it has a profound effect, as you might expect, on colorectal cancers. So again, the different diets lead to different microbiomes. We found some species are associated. Now, we've also found in other studies using visceral fat samples from humans undergoing bariatric surgery that, as I mentioned, visceral fat is a major source of inflammatory cytokines leading to chronic inflammation with obesity. We found that butyrate, that is one of the short-chain fatty acids, dramatically lowers the production of these inflammatory cytokines, but it stinks to high heaven. And so if you take the triacylglyceride form, tributyrate, it's not stinky. And we have an ongoing little clinical trial at the moment that I'm a participant in, just to see what the side effects are. And we're looking to investigate collaborations to look at the effect of tributyrin at reducing the symptoms of inflammatory bowel disease. Okay, just going to end. I know, I'm sorry, my timing's being used up. I apologize. So stop, if I were to give you any suggestions, stop drinking regular pop, switch to soda stream or whatever. We have the best water in the world, Vancouver. Well, don't drink fruit juices. Eat whole fruit, because fruit juices, if they've removed the pulp, they've removed the soluble fiber and resistant starch. Try to eat bananas that have a little green in them. Put the bananas in the fridge. It'll reduce the production of ethylene, which causes the maturation of fruit, including apples, etc., etc. So, not to be racist, but avoid anything white, white bread, white rice, white potatoes, yada, yada, yada. However, if you freeze these and toast them, or with rice and potatoes or pasta, if you boil them, put them in the fridge overnight, reheat them, their glycemic index goes way down because they recrystallize. So that's a very good thing to do. Lastly, next slide. Okay. Okay. Last slide. So just some hints. Go to the next one. So I do what's called intermittent fasting. I eat within a narrow window, mainly to keep my insulin low. You should stop eating at least 3 hours before bedtime, so all that fat doesn't go into your fat, all that carbohydrate. Go for a walk, exercise after eating, 15 to 20 minutes. You can add olive oil or a lemon juice or vinegar to lower the glycemic index significantly. Okay, lastly, I just want to thank the people in my lab who actually do all the work. I have 3 fantastic co-op students at the moment, Vian Chang, Samantha Wu, and Cole Dietrich. And I'd really like to thank my long-term staff scientist, Ingrid Alicia, and research associate, Michelle Yang, who I wouldn't be able to do anything I just talked about today without them. And I'll end the talk there and just mention all this work has been sponsored by the Heck Foundation. Without them, I couldn't have done any of it. Thank you very much. Oh, please stop. Okay.
Thank you for this fascinating talk. I think I have a feeling that we need to bring you back to give another talk for your recommendations in the later half of the talk. We'll discuss that later. Anybody has any questions?
There's a question from Julian.
Go ahead, Gerry.
Please.
Hi, Gerry, great talk. I always enjoy hearing your talks. I'm curious if you could elaborate a bit more on what you think the immune system's role is in the prevention of those metastatic nodules. Do you think it's tumor biology itself changing that allows the immune system to recognize it better? Or is it enhancing the immune cells themselves to allow better killing?
Okay, now, of course, I didn't have a chance to go into more details. So the tumor microenvironment plays a huge role in whether cancer cells survive and proliferate or don't. So the successful cancer cells, the ones we end up seeing in patients, are the ones that secrete molecules that skew the microenvironment to help them grow. So they skew both resident and incoming macrophages into what's called an M2 phenotype. They skew the fibroblasts into cancer-associated fibroblasts. So there's dynamic situation going on. And typically at a very early stage, we feel our optimal diet is preventing the DNA damage to the tumor cells. Once a cancer forms, you likely want a very different diet than if you're preventing cancer. So if you want to prevent cancer, you want to reduce reactive oxygen species. You want to reduce the production of effective CD8 T cells, etc. You want to keep things low to prevent autoimmune reactions, etc., that could lead to DNA damage and cancer. But once you have cancer, you want to increase M1 macrophages and reduce M2 macrophages. You want to increase T effector cells. You want to reduce regulatory T cells, so you want regulatory T cells, which are anti-inflammatory, to prevent cancer. So the scenario changes very dramatically once you have cancer. So, and I want to make that clear. I'm mainly interested in prevention, though we are interested in treatment as well. And you want a totally different kind of diet in prevention versus treatment.
Okay. Thanks for elaborating on that. And on the prevention side, how do you think it corresponds to age and the diet and the effects of the diet?
As we age, there's a lot of things going on as we age. One of the major things is we build up what are called senescent cells. These are cells that normally function. They're given a signal to proliferate, but at the same time, they have DNA damage. They're getting a signal not to proliferate. So they go into this weird stasis mode. As we get older, we get more and more of these senescent cells. And senescent cells are typified by being major producers of pro-inflammatory cytokines. So the buildup of senescent cells causes chronic inflammation. And there's some research going on trying to devise what are called senolytic factors to try and destroy senolytic cells. One way to kill senescent cells is by fasting. So and some people propose every month you should fast for 3 to 5 days to try and rid yourself of these buildup of senescent cells and reverse aging. There's a lot of hype around this. I don't know the validity.
Thank you very much.
I have a question. Is about these geographic differences in terms of access of different kinds of foods and oil across the planet. And is there any epidemiologist kind of studies showing the differences of, because for example, inland people access more carbons than fish and fish oil, and people on the seaside close to the ocean or water have more access to seafood and fish oil. So is there a difference in terms of cancer incidence in terms of epidemiological like data to show that?
Well, because with our industrial revolution, we can now get foods everywhere. Whereas, looking back in history, it's certain there was evidence that people who lived by the ocean, tribal people, were healthier. It's been known that the Inuit, before junk food became available to them on their traditional diet, which is extremely low in carbohydrate because they don't have access to starches. Nothing grows, right, in around the ice caps. And so they had a far lower incidence of cancer than they do today. And when they switched to a Western diet, their incidence of cancer and other chronic inflammatory disorders grew substantially, showing that a low carbohydrate diet has been associated with far better outcomes. Now, I don't know how many are aware of the 1960s huge controversy between Ansel Keys, who was Dwight D. Eisenhower's personal physician, in quotation marks, and John Yudkin from England. John Yudkin said that it was sugar that was leading to the increase in fat and heart disease. And Ansel Keys said it was saturated fat. And so because of Ansel Keys, industry in the 60s switched from fat to carbohydrates, easily digestible carbs. And so that led to it's associated with the dramatic increase in obesity, switch from fats to carbs. And I could go into the pros and cons of all that, but I'll just leave that there.
Thank you. Thank you. Any other people has any questions? I'm sure this is like a day's talk and discussion. There's not an hour.
Yeah.
Yeah.
I'd be delighted to talk again.
Any other people want to have any questions? No. Well, thank you, Dr. Gerald, Dr. Krystal, for the excellent talk and fascinating theory. I'm amazed. Well, thank everybody for attending this conference. Enjoy the sunshine and the weather.
Okay.
Everyone, yeah. And
Bye-bye.
eating healthy food. Yeah, thank you.
Thank you.